Today we'll convert a normal and mutant amino acid sequence into mRNA. Here are our amino acid sequences. For normal, we have methionine, tryptophan, phenylalanine, threonine, lysine, and glycine. For our mutant strand, we have methionine, tryptophan, isoleucine, and histidine. To figure out the mRNA sequence, we have to write down all the possible codons for the amino acids. We will use this codon table to turn the amino acids into mRNA. The first amino acid is MET, also known as the start codon, and it codes for AUG. The second codon is tryptophan, and it codes for UGG. Phenylalanine codes for UUC and UUU. Threonine's codons are ACU, ACA, ACG, and ACC. Lysine codes for AAA or AAG. Our last codon, glycine, codes for GGU, GGC, GGA, and GGG. Now try to find the codons for the mutant strand using the codon table. We already found the codons for the first two amino acids, so let's fill them in. MET codes for AUG and TRIP codes for UGG. Isoleucine can be AUU, AUC, or AUA. Histidine can be CAU or CAC. We are not done yet because there's also a stop codon who does not produce an amino acid. Our stop codons are UAA, UAG, and UGA. Now we ran out all the possible codons, we are going to look to see where a mutation is and if it occurred because of an insertion or deletion. Insertion occurs if there's an additional base added, and a deletion occurs when a base is removed. Pause the video and try to figure out where a mutation occurs. The mutation occurs on the third codon where an A has been added, so this would be an insertion. Since we know our mutation occurs, we could line up our codons to see which codons code for that amino acid. MET and TRIP only have one codon, so we know that those are AUG and UGG. ILE has to be AUU, so it can line up with Phe that starts with UU. Phe has to be UUC to line up with His. His has to be CAC to line up with THR. THR has to be ACU to line up with the stop. And our stop has to be UAA to line up with Lys. And since there are no other codons to compare the normal strand to, it could be any of the lys or gly codons. Normal mRNA is AUG, UGG, UUC, AUC, and any of the lys or gly codons. Mutant strand is AUG, UGG, AUU, CAC, and UAA. However, the strand is no longer functional. And here's a sweet explanation. Here is our normal and mutant sequences. When amino acids are put together, they form proteins. So when you put these chocolate chips together, they form the chocolate bar. Only two amino acids in the mutant strand can form chocolate. Our different amino acids form sweethearts. And when put together, it's not a chocolate bar, it's just a mix of chocolate and sweethearts. Our protein is not functional because we have our early stopped, known as a nonsense mutation, and our frame is shorter, known as a frameshift mutation. So our protein is non-functional because all we get is a jumble of candy instead of our chocolate bar. Here's our normal mutant sequence again, and I hope you guys enjoyed this video.